So I think we're running slightly behind, but I think we should take one question if there is one. Anybody? I'll ask a question if nobody else has one. And maybe actually if any or all of the panelists want to just discuss this really briefly. I think one question that I have is, as we try to really think deeply about how are we comparing the change in phenotypes over time within an organism and relating that to a human disease progression during the life of a diseased patient. There's a lot of causality that happens, and I thought Mary's talk about, understanding, what sort of early embryonic lethalities might exist because we don't have the ability to race our baby pups off to the NICU was really telling. And so how can we do a better job of understanding those causal events that are kind of leading us to interpretations and comparisons that maybe are incorrect? So I think we kind of, all of you kind of touched on that in various kinds of ways, so maybe you could just speak to that really briefly.
I'll start. So then this is purely my own developmental centric thought of this, but I think,
is it working?
No, I think it's working. Can you hear me? No? Oh, okay. I'll be more boisterous. I think that there's a lot that we don't know about development. There's a lot that we don't know about disease progression. I think, some of these associations that we think are, maybe even some that I presented in my talk that I think are compelling and interesting, are going to have to bear the test of hypothesis-driven studies to really determine that. But the systematic possibilities of developing relationships between disease phenotypes that we might see in an embryo and disease phenotypes that might appear in the human population, I think is really untapped. I think there's so much phenotypic information that can be compared, and that trends will emerge that will be supportive of a greater confidence that what we're seeing in a developmental context might be something related to disease. But I think we're utilizing examples where maybe the genetic, or at least I utilized examples where the genetics is pretty clear in some cases. So it's that gene. Now you have to figure out why those phenotypes are, what that means. And from those studies, I think will emerge some rubrics. That's my hope. I don't know if other people have thoughts along those lines.
Well, one thing that Mary mentioned was the subviable class, which I think is an extraordinarily interesting class and has got direct ramifications, I think, for human and clinical genetics as well. The subviable class, where clearly some embryos are dying before birth or around about birth, but some make it through to adulthood and have interesting phenotypes. This is an example of variable expressivity, a tremendously strong example of pleiotropy. Our single gene mutation in the context of development and adult phenotypes can lead to a whole variety of causal outputs that need investigating. But I think fundamentally, and this is the sea change that I think has happened in mouse genetics, is that we have the phenotyping platforms, we have the centers engaged to do all of the phenotyping from development through to adulthood, which I think is absolutely key to understanding gene function and will be absolutely key to making comparisons with human disease as well. So I think that's the message for the future for me.
Yeah, I guess maybe one of the challenges is that most major human diseases are not monogenic, and nor are they static, so that with epigenetics, the expression of genes may change as disease progresses. And that's something that we don't model currently and need to think of how to address down the line.
Okay, all right. I think we'll adjourn for lunch. So thanks, everybody. Let's thank our speakers.